Yeah, let's resume it. Chuck Stevens has one of the most unique ways of presenting. And this is how it works with you, Chuck. I say this affectionately. In a talk of duration n, 4/5 of n consist in Chuck drawing a neuron. Then 1/5 of the talk is all of the content. So I'm going to ask Chuck, don't draw the neuron, show us the neuron, but draw the equations.
Okay.
With that, Chuck Stevens, he doesn't need an introduction beyond that.
Okay. Thank you, David. Okay, thank you very much. Can everybody hear me okay? Oops. That's a test. That actually should remind me to take a pill. But it'll wait. I'm just going to stick it in my pocket. That'll work, I'm sure. Okay, so I'm going to be talking about two brain areas today that use what's called a combinatorial code. And the kinds of computations that these two brain areas use are quite different from the ones that we usually study. And so I'm going to start by telling you what most people, including me, think is the usual way that the brain works. Okay. And the usual way is based on the primary visual cortex, V1. And the reason that's used as an example always is because it's the area that we really know the very most about. And there's an interesting structure in V1 that are called pinwheels, and right here is an example of a single pinwheel, and that same pinwheel is on the left over there in the middle. So there are 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 9 different pinwheels in this piece of visual cortex, maybe that big, and each one is on the order of 1 millimeter squared. In the primate visual cortex, that's you, we have 2.5 million cells underneath every square millimeter, and so that means that this pinwheel over here has 2.5 million cells in it, and those cells are just the same as the cells in all the other pinwheels. So all the information that needs to be encoded can be encoded by each of the individual pinwheels. And if you look at a graph on the bottom down here, let's see if this works. I'm not quite sure where to push. Okay, if you look at the graph down here, this is a graph of a number of pinwheels in the primary visual cortex, and this is a graph of visual acuity, and these are all primates, and we're right up there, and we have about 10,000 pinwheels, so that means we have, in effect, 10,000 pixels in our visual cortex. So each pinwheel is organized in exactly the same way, and here you have lines that are drawn. This line has a slope of 0 degrees. It's flat like that. This line has a slope of 45 degrees. This line here has a slope of 90 degrees, and this line here has a slope of 135 degrees. So anytime there's a line that runs through a pinwheel, the cells that respond to that pinwheel are tuned to match the slopes of lines. And so all the cells, the middle, every one of these circles over here is the center of a pinwheel. At the center of a pinwheel, going up like that, all the cells in that region respond to a slope with a line with a slope of 0. All of the cells along this line here respond to a line with a slope of 45 degrees. All of the cells here respond to a line of 90 degrees perpendicular to this line. All of the cells here respond to a slope of minus 135 degrees. And as you go around the pinwheel counterclockwise like this, the locations along every line here respond to a different orientation. They're called orientation tuning cells, and they are not all of the cells underneath the pinwheel, but there are a very large number of them. So if you have a line or an edge going through this pinwheel here, and you have a line that goes like this, and maybe I can go straight like that, and then maybe it curves up like that and curves down like that. This pinwheel the line going across there, or if the line went across there, the cells up here would respond to it. So in the visual cortex, information about what lines are or other features with other kinds of cells cause particular cells to fire spikes, and information is encoded in terms of which cells are firing spikes. And there are other kinds of neurons that are tuned to things like colors or spatial frequency, and so all of our encoding of the visual world is in pixels. And every pixel has all the same cells in it, and according to which cells are firing in that pixel tells us what the visual system works like. And it's believed by me and others that is the way, that the main way that we know about and the main way that the brain works. But there are several areas that don't work that way, and I'm going to talk about two of those areas now. The first area is the fruit fly olfactory system. The fruit fly olfactory system has three main stages. There's the nose is here. Then there are neurons in the nose, the olfactory neurons in the nose, send axons into a second structure called the antenna lobe. There are cells in the antenna lobe that are called projection neurons. Here's one here, and that cell sends its axon to a circle here. That circle is called a glomerulus, and it collects information from the olfactory neurons that send their olfactory information to that particular glomerulus, and then this cell sends it to the next area of the brain, which is the mushroom body. And these are the places in the fly brain where learning mainly occurs. How many olfactory neurons are there in the nose? There are 50 genetic types of neurons in the nose, and there are about a dozen copies of each type. So all the different odors can make those neurons respond with spikes at different rates. In the example here, there are only 9 neurons, 1, 2, 3, and then the second 3 repeated and the third 3 repeated. In the actual brain, there are about a dozen replications, and there are 50 different types of neurons that respond. And of those 50 types, in most odors, how many odors are? A lot of odors. We don't know exactly how many odors that the fly responds to, but most neurons, most olfactory receptor neurons make most of the 40 cells fire spikes. So this is something quite different from how the visual cortex works, because there you have things according to a property of a neuron, and then that property fires. In this case, you have 40 different types of neurons, but all of the neurons essentially fire to every odor you give the fly. When you get to the antennal lobe, that's the second, this is the first step. When you get to the antennal lobe, which is the second step, the olfactory neurons, the 50 olfactory neurons, send information to 50 different glomeruli. They're circles, they're little spheres of neuropil, which has dendrites and axons in it. And so there are 50 glomeruli. And every one of the neurons of each type sends all of its axons to one glomerulus. And so, in this case, the top ORN here, which is this one, sends all of its axons to a glomerulus there, and there are then 50 types of projection neurons. And the 50 projection neuron types send their information to the mushroom body, where the odors are learned. Okay. What I'm going to be talking about today is basically the first two steps of this, and primarily the projection neurons in the antennal lobe. Now, you can ask me questions at the end, but if there are questions about facts as we go along, just yell out or put your hand up or something, and I'll try to answer the question quickly, and then we can go on. Just in case you have a question about this, you can ask about it. Okay. Okay. So what I'm going to tell you now is work that I did in about 5 years ago, and in that work, what I did is I took data that were published from Rachel Wilson's lab, and those data had been studied, and graphs had been made and so forth, but I had an idea that maybe there was a simplicity that hadn't been discovered. And I'll tell you what I did. Here is the odor recognition slide from Rachel's lab, and along this here, there, and there are 7 different types of projection neurons. There are 50 types. This is a small fraction, but this is what they published. And along here are 18 odors. And those 18 odors are shown twice. One of them is in this color. Those are the ORNs in the nose. And the second one is this color here. And so there it is, and there it is, and there it is. And those are the projection neurons from these 7 neurons going to the mushroom body. I had reason to believe that maybe that these neurons should encode odor information with an exponential distribution of firing rates. Why did I think that? Because I knew that an exponential distribution of firing rates for the odor code, whatever it was, would be a maximum entropy code. And what that means is that information theorists believe that a maximum entropy code is one that carries the most information that it can with the code it has and the number of neurons. And so a maximum entropy code would let you discriminate the most possible odors with the fewest number of neurons. I didn't believe that was true, but I thought it should be true, and I had to test it. And so what I did is I took this graph here, and I made it big, and got a millimeter ruler, and measured the height of every one of the projection neuron things, wrote down the numbers, and there were 126 odors total. And I knew I was wasting my time and it was going to take me a whole morning, but I felt like I had to do it. As I was doing it, I thought, this is just junk. This couldn't possibly be anything. Then I got the numbers together and I rank ordered them according to their firing rates here. And I plotted a graph, a cumulative histogram of the firing rates of these 126 neurons as a function of the cumulative fraction, and it looked like this. It was a beautiful exponential. And so because of that, I knew that I had not wasted my morning, and that this is something that people hadn't thought of before. They hadn't looked at this before. And so what the fly does is every odor. I forgot to mention something. When you go from the nose to the antenna lobe, and then encode the information in the antenna lobe going to the mushroom bodies, there's something that changes. And what changes is that along with exciting all of those 50 types of projection neurons, it also removes the concentration dependence of the odor firing rate on concentration. So all odors have an average firing rate that's the same because of some computation that gets carried out in the antenna lobe. Okay, so what we know is, number one, that every odor on average causes the same firing rates for all of the neurons that do fire spikes. Most of the neurons fire spikes to most of the odors. Third thing we know is that those odors have an exponential distribution that is the same mean for every one. And so the only thing that's different between odor 1 and odor 2 is not the distribution of firing rates. It is not which cells fire spikes, although there are some odors that don't cause certain cells to fire. But it is what rate those cells are firing at. They all have an exponential distribution. If you plot any odor, any individual odor, it's an exponential distribution, always with the same mean. But in odor 1, this ORN, this right here, may cause a very small firing rate in the projection neuron, and this odor down here may cause a very big firing rate. And so the order in which the cells are firing spikes is different from every odor, but nothing about nothing about the distribution of rates or the mean firing rate. No questions. Okay, it's all okay. So this is an unusual feature, and there are more things I could tell you about it because we understand what happens in the antennal lobe. We understand what happens in the mushroom body. We understand how the learning occurs there. We understand how the learning is encoded there. But that's not what we need to talk about today. We just need to talk about other examples where you might have a combinatorial code and ask the question, is this a general rule for combinatorial codes? In other words, are they all going to be exponentially distributed, which is a maximum entropy code, and which would be the most efficient, or are there going to be different things? So far, there are only 3 areas of the brain that have been published, so we know what the code is, and only 2 of those 3 codes have been published. The 3rd code is in the mouse olfactory system, and I can tell you that it works differently, but essentially the same way. It encodes things with an exponential distribution of firing rates, all with the same mean. The more yes, please.
Just a clarifying question. Is it to identify each odor? Do you actually need the values of the firing rates, or just the rank order of which nodes are firing more than which other nodes?
You just need the way it actually works is a little different from that, but you actually just need to know what the firing rates of the cells that are firing spikes are. Yeah. I shouldn't take the time. Okay. So I'm going to tell you about the second thing, and the second thing is the infratemporal cortex has patches of of cells that fire spikes, and there are 6 of these patches in the infratemporal cortex. And Doris Chow from MIT has studied these patches, knows a lot about them, and she published a paper in 2018 that had the output patch, which is the last one down here. So these are in the infratemporal cortex is along here, and these are the cortical folds, and these are the sulci in between. And so this area of the brain has been expanded out so that it's just 2-dimensional. And so Doris studied these face patches right here. Every one of the face patches is different from the other ones, but one of the things that's most interesting about this face patch is that it responds not just to the front of the face, but it responds to every view of the face, including the back, although the back isn't so informative, but all the other views for this patch here, the AM patch, fire at the same rate for a given face, no matter what view you look at. It's view independent. And that is sort of like what the fruit fly does, except it is concentration independent, not view independent, but they're both independent of something that might be interesting. Okay, so I'm going to talk about neurons in the AM. AM stands for anterior medial face patch, which is here, and this is the output face patch, and I'm going to tell you a couple about things about what Doris found. The first thing is she recorded from about 100 neurons, 98. The 98 neurons that she recorded from are shown in this graph here, and this is neuron number 1, and this is neuron number 98 over here, and they are rank ordered across here according to how many faces they responded to. Yeah. Yeah, please.
How did she put the electrodes in there?
How does she?
If you're recording 90 neurons, you have to put 90 electrodes in there.
Well, you record from multiple electrodes, a big patch of electrodes, and you record from multiple ones, but you can separate the different ones by which of the electrodes they're responding to.
What I'm not understanding is, was this somebody in surgery?
Oh yeah. What Doris does is, I'm not describing her experiment very well, but what she does is she does an MRI of the brain. She finds these face patches. She sticks the electrodes right into the face patch. She tests out if that is a face patch and everything. But she knows what face patch she's in and which cells are responding to faces in there. It's a beautiful experiment. Anyway, she showed 2,000 faces to each of 2 monkeys, and these were human faces. And this is the number of faces that caused this particular neuron here to fire spikes. And so about 300 of the faces made neuron 1 fire spikes. And up here, all of the neurons made nearly 100, 98. All of them made spikes. On average, of the 100 neurons, about 80 fired spikes, but it ranged from about 60 to about 90, but on average was that. So this is just shows that this is another case where you have a combinatorial code where many of the faces, basically all of the faces make almost all of the neurons fire. And this is just 2 particular faces. This is face 1 and face 2, and I just plotted how fast each neuron fired for the pair of neurons. And this just shows that they're all scattered around. They don't repeat each other very much. Okay, now what is the face code that Doris uses? This graph here is face 1 through face 2,000, 2,000 superimposed points, and this is the average rate of firing for those cells that fired spikes. So if 80 of the neurons fired spikes to a particular face, that was what it was on average, then what about the other roughly 20 neurons? They were totally silent. So all the neurons, all 100 neurons either fired to a neuron or it was totally silent. And so I took the number of neurons firing spikes at a very low rate or a very high rate, and just averaged across them. And you can see that this line here is an average of 9.4 spikes per second. And you can see that the average number of neurons for the average firing rate for the same 100 neurons was basically 10 spikes per second for all 2,000 faces. So after I did that, I made 2,000 cumulative histograms of the firing of the cells, and this is the firing rate of 0 here. This is the firing rate of 80 spikes per second here, and this is the probability, cumulative probability of firing, and this is 2,000 superimposed cumulative histograms, and you can see that they don't scatter much. They're all pretty much the same. This line here is 2 lines superimposed. If you look over here, this line gets thinner over there. And that's because there are two lines here. One is a cumulative histogram, a distribution function, with a 9.4 mean spikes per second, which is what you get from here. And that is drawn up like that. And then what I did is I averaged the 2,000 histograms point by point going up this way, and I plotted that average, and that's the other line right there. Okay, so the bottom line is that this is exactly the same code that the fruit fly uses, but this is not odors, it's faces, and it's not fruit flies, it's primate face patches. So there is a third example where this is the same code for the mouse olfaction, and there are a number of cases where this may be true, and one of them is in the cerebellum, and David Marr noticed that there was many neurons, many granule cells in the cerebellum fired to every input that you give them. And the reason I'm mentioning this is I don't know what firing rate distribution they had. There's no data that I have for it, but there are half of the neurons in the human brain are granule cells. There are a lot of granule cells, and so it may be that this is not a rare thing that happens, but it's a very common thing that happens. And what my research is concentrating on now is trying to understand more things about this combinatorial code, and to find how many regions there are that I can to find out if it uses the same combinatorial code. So this is definitely a universality, but I don't know how universal universality is, and that's what I'm working on. Okay, thank you.
Yeah. I'm still a little confused about the code.
Yes.
So in the case of the fly eye,
Yeah.
there are,
50
50 times 3 inputs, okay?
Yeah.
And 50 outputs. And what the inputs detect goes out the outputs. And you said that on these 50 lines going out,
Yes.
they're always lit up. Any input lights all 50 up.
Yes.
But what, at different rates?
Yes. Very different rates.
Okay, that's what confused me. So they're all on, but some of them aren't on very much.
They all are on, but some are on at maybe 0.5 spikes per second, and some are on at maybe 100 spikes per second. So there's an exponential distribution, and it's quite a wide distribution. It's always the same mean.
Okay.
And so the code then is which cells are doing it,
Got
or
it.
which cells are doing what rate.
Mushroom body puts the threshold at the mean, above the mean fire.
Okay, so the mushroom body receives those 50 neurons, but then there are 2,000 mushroom body cells called Kenyan cells. Those 2,000 Kenyan cells, each one takes on average a sample of 6 of the 50 inputs and adds them together. And then there is another neuron called the APL neuron, one on each side of the brain. That APL neuron sends its dendrites into the output of all of the Kenyan cells, and it adds up all of the inputs of those Kenyan cells, and then sends it back to the Kenyan cells, all of the Kenyan cells themselves, and this kills the firing of 95% of the Kenyan cells. So only 5% of the Kenyan cells actually respond to an odor, and because of this averaging, it's the firing rate at the extreme of the firing rate at the extreme of the distribution that gets 6 at a time averaged together, and then those neurons, which we call a tag, and that tag is sent to and does the learning, and it does a different learning for different odors, and they're all different. There's some that are the same. So methanol and ethanol are very similar for us and for them, but we and the fly can both learn to tell them. Apart. But in general, any two average odors, and there's no overlap in that tag.
I think Pablo is next, and then.
Thank you, Chuck. I was wondering how you explain the constraint on the average rate and the connection with the maximum entropy distribution. If you just know the mean of something and suppose that the neurons are constrained to fire on this average rate,
yeah,
the prediction, the maximum entropy prediction would be an exponential distribution.
It is, and that's what it is.
Right, so but the important point is why they fire at a kind of a average rate of 10, or if that varies or not, or...
No, not every neuron fires, but most of the neurons fire for most odors. There are a few neurons that only fire to 1 or 2 odors, but that's extremely rare. And in general, if you just take 2 odors at random, they will fire with an exponential distribution that is identical. You can't tell them apart.
But how you explain the average rate to be fixed at 10? Why is fixed at 10? I mean, why the average rate of the different neurons is the same?
Why is it?
Yeah, the first graph.
That is the average across the 100 neurons that are firing spikes to that face.
And what is the explanation of that constraint?
No, we don't. I mean, it's made to be that way, but I don't know how it achieves it in the circuitry.
Okay.
I don't know how it does that, but it does.
Chuck. Chuck.
Oh, sorry.
That's okay. John's going to ask a question too. 2 related questions. One, I think is sort of related to Pablo's, and that is the first one is what is the advantage of this combinatorial code? Is it really the kind of efficiency thing you started with, in terms of the number of neurons? And the second question is over, say, other codes. And the second is, do you have any sense of how hard it is for the brain to discover it?
To discover?
Discover this as a solution.
No, I don't, but I know other things that are informative for that. So I have also studied the output for the ORNs. And those, everyone has a different mean, but they also have an exponential distribution of firing rates. And so somehow built into the ORNs is this automatically an exponential distribution. It's just that the rates are different. But then the rates are removed at the first step, and so that's how you end up with an exponential distribution, all with the same rates. I think I forgot your second.
The first question was about the advantage of the code.
Yeah. Okay. So the advantage is that if you have a combinatorial code, and there are many possible combinatorial codes, but this is a very simple combinatorial code. It's something that's apparently easy for the brain to do because it can make those distributions easily. And so that would mean that if you can do that, this would give you the most number of faces or the most number of odors for the smallest number of neurons, and that would be the advantage of it.
So it's efficiency.
Yeah.
Yeah.
Okay, sorry.
Am I next? Yeah. A comment and a question. The comment is that Bialik's group at Princeton has worked with rats going through mazes
Yes.
and shown that the pattern of correlation firings in neurons is also a maximum entropy distribution.
Yes, I can believe that.
Yeah. Now the question is the perceived intensity of the odor related to the firing rate, and does the exponential have anything to do with the logarithmic perception of intensity?
Now, I left out something, and that is there's another area of the brain which also is the olfactory system, and that one is also very orderly, but there's not learning there. And so presumably this hasn't been studied, so we don't actually know, but presumably the intensity of odors is there, and this is where the learning odor happens.
Okay.
Yeah.
One last question, Chuck.
I'm just going to.
In V1, you talked about sort of the edge-sensitive neurons in V1.
Yeah.
And the sort of simplified story of visual cortex is that it's hierarchical, that the first edges are detected, and that information goes to another layer in which more complex shapes are detected that are made out of those edges, and so on. And so there's this hierarchical structure because the visual world is somewhat hierarchical. Is there an advantage of this different code that V1 uses because of that hierarchical structure?
No, they're about the same, but it's not different. It's not different layers of the cortex. There are about 36 different cortical regions, and each of those cortical regions responds to some specific feature of environments, like motion detection. And so there are motion detection neurons in a region called V5, and those motion detections don't care what it's motion of. It just looks at the rate of motion, and it knows what the rate is, and it knows which direction it's going, but doesn't care about anything else. And we don't know what all of the areas do, but there are speculations about the different regions of the brain do different things, all with versions of the output from V1 and sometimes from other areas. Okay. So